Yeah, so thanks everyone for coming and attending the Science on Tap. I hope you have fun and that you have your drinks with you. So today I'll talk about imaging fast hemodynamic responses with high spatiotemporal resolution fMRI. This is some work that I've been doing here since June with, in the group of John Palimeni and Laura Lewis. Micah asked me to tell a little bit about myself. So my background is in physics engineering. I come originally from Brazil, so that's where I studied. And then I moved to Germany, where I wrote my thesis in imaging tissue susceptibility. And then I started my PhD in a collaboration between Siemens Healthcare, working with the EMR scanners manufacturers, and the Donders Institute in the Radboud University in the Netherlands, where I worked mainly developing sequences for imaging fMRI, in particular fast fMRI. So perhaps most of you have heard already of multiband accelerated sequences, and this is mostly what I worked on during my PhD. Without further ado, I'd like to, maybe I should have called this talk A Tale of Vascular Dynamics and Phosphol Oscillations. And I'd like to tell you why we think it's interesting to image fast hemodynamic responses and why we're interested in high spatiotemporal resolution fMRI. So first I'd like to start with a sentence that I came across recently when I was looking at this paper from Peter Bendettini in 2014, where he writes that each individual, each task, each time series, each voxel, and each subtool factorization contains extractable neuronal and physiologic information when we're talking about the fMRI signal. And he asked the question, how do we actually extract that information? And so here I'd like to maybe extend this and say, not only how do we extract this information, I don't promise that I'll give this answer today, but rather what is this information that can be extracted, and what is the way that what is it that we need to do in order to be able to answer this question? How do we extract that information? And so I like to start out by talking a bit about some fMRI studies that I've been doing that will lead to this idea of imaging fast responses. And I would like to start at the beds of the calcareous sulcus, because this is the region that we call V1. That is the primary visual area. That's what we usually like to test when we're talking about fMRI responses. And so traditionally we've imaged with voxels that are more or less this size. So we place it on the beds of the calcareous sulcus, a 3 millimeter voxel, and we could play a flickering checkerboard, for example, and we would get a response to look more or less like this. This is what we call a hemodynamic response function if we're imaging with fMRI. So we play our stimulation and then we wait some time, some 6 seconds, we would see a peak in the activation that we measure, or the change in the signal that we measure in the beds of the calcareous sulcus. And then after a certain time, the signal decays again and goes back to baseline within 20 seconds. But if we look closer into the beds of the calcareous sulcus, let's first draw a set of surfaces over it. Here I'm showing the white matter surface in blue and the pile surface in yellow. And if we zoom in right into this square here in red and we look closer at it, if we zoom even closer on it, sorry, the presentation is hanging, I don't know what's... and we were able to look into the microscope, we would see that within these 3 millimeters that we're imaging, there are differences in the circuitry across the different depths from the white matter surface to the pile surface. And there are also changes in the vasculature. If we were to look at it in an electron microscope, we'd see that the vasculature changes across depths. So here in this image, we see that the blood would come from the pile arteries through the penetrating arterioles into the capillary beds, closer to the white matter or closer to the deep regions of the human cortex. And then it would be drained again, this blood would be drained again to the surface through ascending venues. So there's also vasculature changes across depths. And because, of course, the vasculature and the neurocircuitry changes across different regions of the brain and across subjects, we would expect that canonical hemodynamic response that I showed earlier to also depend on the brain region, on subject, stimulus use, unconscious tension, of course, on the micro and macrovasculature, and on imaging parameters. So the question is this true? And is using such an idea as a hemodynamic response the way to go if we want to one day have fMRI as a tool, as a gold standard for measuring neuronal and physiological information, as Bendettini Estes perhaps or proposes to do in his paper back in 2014 already. And so, of course, we have reasons to believe this isn't true if we consider what I've just mentioned, right? But how do we go about actually investigating whether this is true or not? And so first I'd like to now transition into the fast hemodynamics and tell you of one experiment that challenged this idea. And I think more than only looking at fast hemodynamic responses, which I'll mention in a bit, it's interesting because it's really opens up all this idea that the way that we look at our data may allow us to extract even more information than we previously believed. And perhaps this idea of constraining ourselves to a model that has strong assumptions on what the signal should look like may not be the way to go for the future. So this canonical hemodynamic response, let's look at it in its response function. If we look at what the flickering checkerboard and we look in the response, and if we look at the frequency response, we would see that if this flickering, if the modulation of this flickering, if we were to show the amplitude of this flickering at different frequencies, here 0.1 or 0.2, 0.3 hertz, we would see that the response that this hemodynamic model would predict would decrease. It would be about 10 times weaker at 0.2 hertz than if it was 0.2 hertz, and it would be 5 times weaker at 0.33. And so, to this flickering checkerboard. And so what Laura Lewis, my mentor, and John and others at the Martinos did a couple of years ago was to actually try and see if this is true by modulating this flickering checkerboard with different frequencies, and then looking at the actual response that they would measure, and just to investigate whether this would follow the predicted hemodynamic response. And so here in this figure, we see the stimulus in purple, and then its response, and we see that the oscillation amplitude that would be predicted should decrease as the model would predict, but the data that they actually measured showed that the responses are much larger than previously believed. And so when we think again about the microvasculature and macrovasculature, and we observe that even at a resolution that wasn't extremely high, we can see amplitude of responses much larger than predicted, it begs the question, is there some mechanism behind the vasculature that accelerates as stimulus get faster? Or what can we explore within the helm of fMRI to get this information that is not there? How do we break apart from this model and have a more clear biological understanding of the imaging that we're doing? What is the relation between the stimulation, the micro and the macrovasculature, and the fMRI signal in order to extract this neuronal and physiological information? So if we look again at the beds of the Cochrane sulcus and the hemodynamic response, but if we were to look at it across depths, then we could ask, is this response, how does this differ across these different depths? Now, if we have a very fast stimulation and we think, for example, that these two regions are being stimulated at the exact same time, then how does this signal drains to the upper layers, or is the signal then different? And so what I've been doing in the past year or so is imaging these oscillations, these fast hemodynamic oscillations in V1, and trying to look how the amplitude and the temporal specificity of the signals varies across these different depths. So here's an example of an image that we acquired, the activation, and if I project or generate layers on top of this depth-dependent layers on top of this activation, and look at how this oscillation or the amplitude is changing across depths in function of the frequency. We could see in this image, for example, that the frequency, if it's slow, that the signal is much stronger than as the frequency gets smaller, of course. But what's interesting from this figure is that there's essentially that the signal gets much stronger at lower frequencies in the pio matter than it is close to the white surface, in the pio surface than white matter surface. And this is not, the effect is not as strong with higher frequencies, suggesting that there's also a nonlinearity in there, right? That this draining of the signals, for example, that at a low frequency that the signal drains and ends up accumulating in this pio surfaces, whereas this doesn't happen for the fast signal. So there's a phase cancellation effect. Right, so if we look at the activation here, and we consider the time that blood is taking to drain to this different region, as the stimulus get faster, then this distance starts to matter. And so we would see differences in the pio surface that are only related because of the differences that it takes for blood to drain from these different regions. This is why that nonlinear effect. And so looking at it again, we see that as the stimulus frequency increases, that the ratio of the amplitude of the signal that we get across layers tends to be closer to 1. And when we have a lower stimulus, that the intensity of the signal in the pio surfaces tends to be much, much larger. So this is just, it's not necessarily an effect of the canonical that would be explained at all by the canonical hemodynamic order that we see, but it's truly something that is underlying and is only observable as we move towards these faster responses and start to look at stimuli that deviate from the common assumptions of fMRI. And gaining an understanding of how this nonlinearity happens and how the signal changes, these fast signals changes across layers, could potentially allow us to correct for these differences and then infer more quantitatively or more specifically what is the signal that is coming from each capillary bed, and perhaps one day even potentially think of causality or the time and the amplitude of signals across different layers of the brain. Yeah, so here's another image. I apologize that it's slightly blurred, but here is measuring the time differences. This is a histogram. If I took all the activation at the close to the pio surface and all the activation close to the white surface and just plotted it as a histogram, this x-axis is showing the time with respect to the stimulus. So is the HRF that I measure to this oscillating stimulus, what is the delay in the white, close to the white surface, what is the delay close to the pio surface? And if I measure this difference in time, we see that the signal that we're measuring from the deeper layers to the exact same stimulation is faster than the one in the pio surface. This is also something that we would not have picked up if you were thinking exclusively on analyzing our data using a predefined canonical model, for example. So here again is more or less the same data, and what I'm showing here are different runs for different subjects acquired with these two different frequencies. And here I'm plotting this delay that we measured in the pio surface and in the white matter. So everything that is below this dashed blue line means that we're observing signals that are happening slower in the pio surface than there are in the white matter. And so we see that as we tend to get slower stimulations, that the signal drains and is much slower at the pio surface, this difference is increasing, suggesting that as we increase the amplitude, there's an effect not only on the amplitude across these depths, but also under temporal specificity. With faster signals, we get signals that are closer to the time of activation. So we increase the specificity of the fMRI signal. And this may have very interesting implications for looking at things such as naturalistic stimuli that are obviously happening much faster than these very strong and slow stimuli that we used before. So looking at the fast fMRI is not only to allows us to look at faster responses because of faster responses by themselves, but really to understand what are the mechanisms behind fMRI responses in general, how do we model this in the future, and how will we eventually separate the neuronal and the physiological information and get a true understanding of the hemodynamic signal that drives our fMRI studies. And hopefully one day fMRI, if we do have an understanding of the signal across depths and with respect to different neuronal circuits, we could use it even as a gold standard to look into circuits in the brain. So what we've shown is that there are differences in both the temporal and spatial specificity of signals across depths. And to do that, we used fast stimuli to investigate these differences. That these faster stimuli are possibly more specific to the site of activation. And we argue that this is the case because we look at the time and the delays between the actual stimulus that we play, these oscillations, and the response that we measure, and that understanding these effects may allow us to eventually better model the fMRI data and better separate neuronal and physiological information, and potentially look into causality with fMRI. And so, yes, how do we extract this information? I would argue that we will extract this information by getting a better understanding of the signal. And how do we get a better understanding of the signal? By pushing beyond our assumptions and look at how the signal behave in edge cases such as FAST at high resolution, both temporally and spatially. And yeah, I guess this is what I had for you guys today. So thank you.